We talked about on Tuesday about how they went about to sequence for the first time an ion channel, and let me just remind you, it was based on this idea that technique, or series of techniques where you take a protein, you purify it, and the sodium channel was a very good thing because for a long time we have known about prototoxin and that it's specific to channels that are involved in generation of action potentials, and in particular in the rising phase of the action potential. So, good candidate to be brought into a sodium channel. And so, the only thing you needed is a good animal that had a lot of sodium channels in some organ that you could harvest and then purify this thing by somehow labeling the prototoxin, and then you just look for where the label is and somehow purify it. And that's biochemistry and but then once you have a little chunk of the sequence, and hopefully several chunks of the sequence, you use the genetic code that we know now, we knew at the time already, and how it codes the sequence of nucleotides of DNA or RNA code for a protein, for the amino acids. You could guesstimate, guess of what the sequence at the DNA level would be given the protein sequence that you have. But that's a lousy guess, right? You know, and I hope you remember that the codons that code for each amino acid are not just one per amino acid. There's a little bit of floppiness there, and so it's a guess. And with that you start. It's a good guess, so you get good. And what the idea was is that you synthesize a little bit of the sequence of nucleotides artificially, and you can do that based on what you have learned about the protein sequence. And with that, you now go fishing. You really you use it as a fishing bait to grab bigger pieces of RNA that may have that sequence in them. And that may be many. Let's say you have 3 proteins in your genome, or in the fly or mouse genome, or whatever this was done first, that have the same sequence. So you will have 3 proteins. They won't be all exactly alike, although that little piece will, or maybe isn't, because of this vagueness of the coding that we just made that piece of nucleotides, build that piece of nucleotides based on, right, the amino acids are coded by different codons, and therefore you don't have exactly the sequence that your sodium channel or whatever you're fishing for has, and that's why you're going to get other products. But you do that with many little pieces, hopefully covering the whole length of the sodium channel. And so you get, with one little fishing bait here, you get some more molecules, with another one that is made to another region on this protein, you get others. And then, little by little, once you have a lot of these, those that correspond to one molecule will have a lot of overlap between those little pieces. So, let's say this is your, and we call these primers. So you have primers 1, 2, and 3. Let's start with 3. And this one catches a piece of RNA, and remember, you take the RNA from the cell, and from that you synthesize back a piece of DNA that we call complementary, or cDNA. That's what you work with. Now you have a library with a lot of this stuff. You make any copies. And this primer somehow matches this little piece of that molecule, right? And so it fishes that out. You need 2 primers to that. And then this one, let's say, fishes out a piece like this. And this one fishes out a piece like that. If you're lucky you have a great one or whatever, but you haven't. And the likelihood that different proteins that may have been, or sequences that may have been extracted by 1 primer, by 2 primer, the likelihood that they will have extracted the same one is there only if this one codes for the same protein. Let's say that. But the idea that if you have many different pieces, let's say this one extracted this, and another one, and maybe 5 of these, okay? whatever. But only this is the sodium channel. Okay? These are all similar here, but they're not likely to be similar everywhere else. Likewise, with this one, you may have many, right? But these are only going to be similar in that region. And maybe in here too, you may have many. And they're only going to be different or similar in that region. But once you overlap them, you'll know that those that are not in sodium, this is a sodium channel, this will be similar to that. Sorry. No. This will be similar to this. These will be similar to that. But this overlapping region will be similar to each other only if they are the same, if they are sodium channels, if they belong to that same sodium channel. A different protein will likely have a very different region where these things overlap. Is that clear? That's it. Anyway, I don't want to dwell on it very much, but that's how these people extracted a 4 long length of the entire cDNA that goes to the sodium channel that turned out to be 2,000 amino acids long, very long, deep, and they then started looking at the properties of those channels, and they found that there were these little stretches that were non-polar, and therefore likely to be stable inside the membrane. That was the logic, that was the thinking. And so if you have a membrane like this, what they found is that there were enough of these so that you could imagine, and these non-polar regions were separated by polar ones, and you could imagine then that the polar ones were happy in an aqueous solution like the intracellular or the extracellular medium, where you have ions and a lot of water, all polar molecules, but the pieces in between transition through the membrane and they cross the membrane like that. Now we know that these regions actually form alpha helices, so we knew a little more, they knew a little more about the structure of the protein, because that's probably the best way of stabilizing a piece of protein in non-polar environment. Because even if you have charges on the side chains of the amino acids, they may be neutralized by adjacent ones, or even within the alpha helix, some of these may be neutralized by adjacent side groups. These are charged, but not necessarily charged, even just polar. They may be stabilized by interactions nearby, within the same molecule or with adjacent molecules.
You mentioned that there was a way to stabilize a protein in a non-polar environment. Can you repeat how that's possible?
Yes, this is what I was just saying. So either the whole chain of amino acids within the alpha helix is non-polar, and you have glycines, for example, where the group is in the hydrogen, or amino acids as well, but there's a whole bunch of them that have side groups that are not charged or polar. Okay? So either you make the alpha helix with that, or if they are polar groups, they are in positions where they neutralize their charge or polarity between each other within the alpha helix or with an adjacent alpha helix that may have a group like that. So it stabilizes them side by side. That can be good because that makes the whole molecule stabilize itself with these intermolecular interactions. Like that. Okay. All right, and so they found that we have these transmembrane segments, and we call them S1, S2, S3 for that reason, segment 1, 2, 3, 4, etc. And then they found this little hair, or they proposed this little hairpin-looking piece of DNA, protein that crosses the membrane but not all the way, followed by another segment. So this is segment 5. And this is the so-called P loop. Okay, and in the sodium channels, what you find is that this repeats itself 4 times. Okay. More, the whole thing, 4 hairpin states, and then again. structure of this protein network very similar between these domains, and for that reason we call them homology domains. They're given Roman letter that is 1, 2, 3, 4. And we start seeing it happen. All sodium channels look like that. Moreover, all calcium channels also look like that. Very similar. So this is the general properties of sodium and calcium channels. They all have 4 homologies. I should probably say most. Think of it now. The voltage gated calcium and sodium channels, I believe they all very much look like this. They have 4 homology domains. They have 6 transmembrane segments in each domain. And they have this little hairpin looking thing that crosses the membrane only partially, that we call the P or core. That's what the P stands for. And we'll talk about that in a second. So that's the core or P. Before I talk more about this, another strategy to extract to sequence channels was used for potassium channels, and using flies. Flies, and you may know, Drosophila is a well-known animal for doing a lot of genetic studies, because they use a lot and fast and so on, but also because you can find lots of mutations in the natural population, or you can induce them with chemicals. Just irradiate them with UV light, or you put some nasty chemicals in there. They reproduce fine, but then they start producing little changes. One of those changes was a behavioral change that you couldn't tell until you anesthetized the fly. And the way you anesthetize flies is you put them in chloroform, jar with chloroform, cover them, and put a little cotton, soaked with some chloroform, and the flies will just go. You take it out and they recover just fine. And you use that to sort them, for example. You're looking for a mutant, let's say, different color eye. They tend to call it, the eyes tend to be white, red, and all those mixes and so on. You need to look at them, so you mobilize them like that, and then you sort them. Under the microscope, you will see a guy with a little brush. So this way, that way. Then you have your populations, you wake them up, and you bring them again, and you can produce a pure population now of red-eyed Drosophila flies. In one case, what these guys found is that instead of the fly just going limp, it started shaking, its legs in particular, which is not the way the fly goes to sleep. So they called it a shaker fly. And this shaker fly turned out to have a mutation in a piece of the chromosome, and I forget which one, there are only 4, in a piece of the chromosome where you have lots of concentrated mutations that tend to happen more often than in other regions. You call that a locus. And that locus was called shaker because mutations, and there were lots of molecular biological techniques or even genetic techniques to find out where these loci are in the chromosome. More than one mutation would produce that behavior. So because they were localized in that region, people thought, movement of legs, and they have something to do with ion channels, right? Because action potentials are needed for you and flies to produce a movement or a leg. Maybe there is a channel involved in here. A lot of these mutations pointed to that region in the chromosome. So people extracted that piece of the chromosome, and again with enzymes and stuff that you can cut DNA with, and you started sequencing it. And within that sequence, what they found was something that looked like this. Again, doing the plotting the hydrophobicity of the amino acids and so on, what they found is that, and then translating it into a picture like that, they found this. What does that look like? Doesn't it? Looks like just like one of those homology domains, right? But it was short. It was just one piece. It was 500 amino acids, 700 amino acids long, something not very long. And with 6 transmembrane segments, one thing that and yeah, okay, so they synthesized, they came up with this structure. Now they had the sequence, so they can synthesize proteins in a dish. And the next thing you do is you want to know what is this, or what is that, what does it do, right? So here we're saying, I said channel, but how did I know that this was a sodium channel? For now, we only have the picture, right, and hydrophobicity plots and stuff like that, and ideas, but nothing really concrete. So the way you do it is you make a protein, or make RNA better. You just make RNA out of the sequence of this long cDNA piece that you extracted from these experiments. Now you make either mRNA, which is kind of difficult to make, it's very unstable, but it's doable, and then you inject it into a cell and see what it produces. So if you inject a lot of it, you should produce a lot of the protein that it codes for, right? Makes sense. The way to do it, or the way it has been done, is using frog eggs. And have you ever heard of the xenophus frog? I'm sure some people have. No? The African clawed frog, which has escaped and has a lot of trouble because it's voracious and very aggressive. Well, a lot of biologists in general use that frog because it produces lots of eggs, and very often. And you can take a female, you open a little slit in their belly, take some of those eggs, slow it up again, you leave it there, leave the frog in the tank again for another month, it will produce more eggs. And it produces so many that you can do whatever you want. You don't know what to do with so many, typically. And they're large. They're about 1 millimeter or so, up to 1.5 millimeter or so in diameter, so you don't even need the magnifying glass. All you need to work with these. And then you take one of these pipettes that I've shown you before, make the tip very broad, poke the electrode with it, and just push with pressure a solution that contains this mRNA into the cell. Let it be there for a day or two, and then see what is the cell doing. And if it's a molecule, a protein that produces color, the egg will be colored. But if it has an ion channel, it should be able to measure the current, right? And this oocyte otherwise has very small amounts of all different kinds of currents. But if you measure it when you record, it's something that looks, and I'm going to plot this IV plot, as this is called. What they saw is something that looks like this. So the egg itself had currents that looked maybe something like this. So at high voltages, it produced a little bit of outward current. In an injected egg, see here, in an injected egg, what we saw was this huge current and negative. What is a negative current? And moreover, let me tell you something else. This was about +60, in the neighborhood of 50 or 60 millivolts. What does that suggest to you? They're not working? Why? This is current. So they're passing current. That's what a channel does. But it's negative. What does negative mean? Potassium? So he said no. So remember, when we have sodium flowing in, what do we call that? Yes, it produces a depolarization, but here's this is the oxygen. It's negative. Switch your logic. 50 years ago, when people studied how the natural potential is generated in the squid, they called this a positive current. Well, for many reasons, people decided that's not a good way of calling it. I don't want to know. The positive current will be current that gives positive ions. And the current that puts positive ions in is going to be a negative. Calcium and sodium, which are very concentrated outside, carry positive charges into the cell. We polarize the voltage, yes, but we call that a negative current. Just remember, it's convention. It's nothing. There is a certain logic to it. So what is this?
Negative.
Negative. What is it likely to be?
So,
Carried by it, right? Huh? Why sodium?
well,
Because it's negative. One. What else?
negative, and it shares similar domain.
No. We don't know this.
We don't know that.
This is how we found out about that. I just jumped the gun there. This is what you have to do first to find out what is this protein that looks like that, or we are proposing looks like this when you stretch out the membrane and look at it like this. But we don't know the function. We know a bunch of things. We know that we did this whole thing about extracting the proteins and sequencing and all that using CPX as our purifying agent, which we suspected is a sodium blocker, the sodium channel because it blocks action potentials and all that. That's one thing we know, and then we know this, but that doesn't tell us anything about its function. Right? But then you put it in an oocyte, and this is what it produces.
The 60 millivolts.
What does the 60 millivolts tell you?
It's reached its bound.
ENA, right? That's about where ENA is, the equilibrium potential. So that's the telling you. Now, what you do, what you do to confirm this? What would you do to confirm that this is sodium channel? Okay, so maybe you have calcium channels, calcium ions that also have a reverse potential. It's positive, and you're really certain. What do you do? What do we know about this? How do we start? How do we start this whole process until we got to this? What was the first thing that I said today? Find the channel. Huh? Yes? No, before this. Before. How do we start here at the very beginning? Yes, what about it? No, before that. What did they use to purify the protein?
PCR.
Before that. How did they purify the channel? The protein, before they sequenced it. Using the toxin. Remember?
CPX.
That's how they fished out the proteins to purify it. So what should PPX do to this? Should it do anything to it? It should flatline it, exactly, right? Because this is a sodium channel that was supposed to have been sequenced based on the fact that the prototoxin blocks it. It better block it, otherwise it's not the same thing. But that's exactly what it does. It does block it. So channel, you put enough of it, it will block it. What else could you do? And you really want to make sure, so you do a lot of things, not just one.
So on the graph, the blue line, is that the normal sodium channel?
No, that's the normal current that the egg will produce without any injected external RNA.
Okay.
So what is it likely to be mostly? Just remember, a current like that shows how the current varies with voltage. And if you're not doing anything else, it shows all the current up front. So it's a sum. Well, the resting potential is not shown here. I didn't draw this very clearly, but the rest. How would you tell what the resting potential is in this graph? That plot there, right? So except that here we're talking about the whole stuff. And that's why these curves are smooth and so on. But so if I'm drawing this, how would I tell where the resting potential is? What would I need to do? What would this trace need to do?
Across the.
Yeah.
Right? This would be expressive potential. This tells you that there is more outward current flowing at positive voltages than negative current is probably at negative voltages. Away, I mean, we move away by the same amount from the resting potential, right? Which is the reverse. I mean, it's the point where currents don't flow. So you have the driving force will be the same. You move the same distance from here to there, this to there, except for the sign. That's what you need to compare. So more current is flowing outwardly than it seems to be flowing inwardly at equidistant points. But there isn't this. I mean, the important thing is that this red trace you don't see it in the normal Hall sight. Okay? You only see it once you injected this cDNA mRNA that you make out of the cDNA that you sequence. And you put a lot of this in there, make this a very big current. I'm sure that something is happening. Okay, so we established that PPX should block it. So in PPX, this should be like this, with an mRNA injected and no PPX like that. We established that this is very close to the reversible equilibrium potential for sodium. What else could you do to make really certain that this is a sodium current? Obvious. I mean, it's simple. It's not. I'm not asking for something very complicated. What carries the current? So, what could you do to make sure that this is the sodium current? Huh? But you can't already show that it blocked it. And there's always the possibility that PPX is blocking something else, right? Since channels are defined by what ions flow through them, we'll make sure it is a sodium channel, not a current that's blocked by PPX.
Calcium switch?
Huh?
A calcium switch assay?
Calcium switch?
Or just put it into calcium to see if it responds.
Okay, yeah, put it into calcium. Saturated calcium. I mean, there's calcium already. You mean increasing calcium concentration?
Yeah, saturated calcium concentration.
What would you expect to see?
I wouldn't expect to see it. I would see, if anything, you would see it, making out a point.
Okay, so good point. You put calcium, put a lot of calcium, what we see is no change. Good, that's a good indication. But let's now go for the confirmation here. Calcium doesn't do it. What about, there's magnesium in the water too, with the magnesium. You don't want to make sure it's sodium. I mean, if that's your hypothesis, right? You're along the right track. Well, what would you change? Sodium. It's a sodium channel. You remove sodium, let's say. It shouldn't be flowing, right? Exactly. So, let's say your egg has, I mean, this thing is sitting in a bath which has a lot of sodium. Now you reduce it and make this even smaller than that, let's say. Okay? Or make it 0. What should happen? These channels, by the way, the gating of these channels does not depend on the sodium, on the ions that it carries. None of these channels gate differently if the concentration of the ions that flow through it has changed. The gating is a property of the protein, not of the ions. Okay? Right? The gating is not a property of the ions that flow through the channel, but it's a property of the protein that is gating the ion channel itself. So, want to make sure it's a sodium channel. What would you expect to see? Let's say you put this, instead of 0 sodium, okay, 0 sodium, what would you expect to see? Huh? So you change the voltage, your experiment is, you put the cell at this voltage, at that voltage, at that voltage, and so on, at all these voltages. This is your experiment, testing all these voltages. What you got is a current that looks like this. So at this voltage you get this, at that voltage you measure that, at this, and so on. Now you put it in 0 sodium, what would happen? Okay. Who votes for a straight line? One vote. Okay, who votes for something else? Nobody's voting. Nobody's allowed here. Huh?
It's not equal.
No vote or no, I don't know.
Would sodium move out and be
No, who votes for that? So one alternative is this thing becomes flat, like just like adding TTX. The other one is the channel, the current now looks positive because sodium instead of moving in is moving out. Okay, vote for flat. 1. Vote for out. 2, 3, 4, 5, 6, 7. You lose, Sarah.
If we take the sodium out, this is nothing, you just said right that because heating is a property of the channel, would the channel still be open, so it would just be that anyway?
Yeah, but this indicates the direction of the flow, right? That's why it's inward, because it's flowing in. But now you have more sodium inside than outside. You open those channels, the electrical, the diffusion would just prompt it to move up. That is the correct answer, and what you would see is a tiny current probably, okay, but it would be upward. All right, excellent. So that's how this was established, and now we can do this with eggs, with these xenobit eggs. For that, you have to make mRNA and put it into the cell. These days, you can do it with mammalian cells that are immortalized cells, with tumors and so on, that can be propagated forever. You divide, they always remain the same. Mutation here and there, maybe. And those are much easier to deal with, especially if you're dealing with mammalian channels. You just inject the DNA. You need to inject it. You just shoot it, and there's even flat call this the DNA gun. You literally shoot these things into the cell. And you will see exactly the same thing. Okay? So sodium channels do this. If you look at the trace, so here you have the voltage, you polarize the cell, okay? And when you measure it, so it's sitting at wherever it is current, the voltage, the channel will do this and turn itself off. What do we call this? Inactivation, right? The channel activates, this is activation, and then it inactivates at that voltage. Okay? It's just a